Most people are familiar with the existence of carnivorous plants, whether it's from a Nintendo game or an off-Broadway show. A carnivorous plant is defined as a plant which derives at least part of its nutrition from eating small animals. They attract these animals, which are usually insects, with sweet-tasting nectar. Then, while the animals feed, carnivorous plants capture them using slippery, waxy pitfall traps. But how and why did plants evolve these structures? A lab in Japan wanted to find out. They chose a carnivorous plant called Cephalotus, which grows both flat and pitcher leaves. Then, using genome and transcriptome sequencing techniques, they compared the genetic information of the leaf types in order to identify changes responsible for carnivory, including genes affecting prey attraction, capture, digestion, and nutrient absorption.
We're all affected by temperature on both a conscious and subconscious level. Consciously, we may cover ourselves in a blanket if we're cold or fan ourselves if we're hot, and subconsciously we'll sweat if we're hot and shiver if we're cold. But cephalotus respond to temperature in even more strange ways than we do. A group of scientists wanted to see to what extent temperature affected cephalotus, to see if there was a temperature-based reason the plants sometimes express a pitcher leaf and other times express a flat leaf. They placed the cephalotus plants in two different conditions: 15 degrees Celsius to mimic late fall and early winter and the loss of most insects, and 25 degrees Celsius to mimic summertime when most insects are flourishing. What they found was that in 15 degrees Celsius, the plants would transcribe the genes for the flat leaves so that they could get their nutrients from photosynthesis, while in 25 degrees Celsius, the plants would often transcribe the genes for the pitcher leaves to get their nutrients from the insects it would catch from its slippery surface.
As Tyndall said, the morphological development of these leaves are highly temperature dependent. So to study this, the lab in Japan took samples from leaves and pitcher plants and took the entire mRNA profile of each and analyzed it. Sequencing the mRNA and relating it back to the genome can give a profile of what genes are being expressed in each plant structure. It was found, not shockingly, that the flat leaves contained a higher proportion of photosynthetic genes than the pitcher structures. Additionally, the pitcher transcriptome contains a larger amount of morphogenesis genes than the flat leaves. This is most likely because of the structural complexity of the pitcher structures. These genes are most likely controlled by thermoregulatory factors and contribute to the thermoselective expression of pitchers at high temperatures and flat leaves at lower temperatures.
Cephalotus follicularis is a carnivorous plant that evolved from non-carnivorous uniquat plants, with a close relative being the coffee plant. In order to determine the way in which the evolution of predatory systems occurred, follicularis' digestive proteins were sequenced along with 3 other distantly related predatory species. This allowed for them to not only examine how carnivory could independently evolve from non-carnivorous plants, but also the way in which carnivorous plants continue to evolve and differentiate in, for example, trapping methods. When they examined the digestive enzymes utilized by all carnivorous plants, they found 35 similar genes that produce the same types of proteins used in digestion for all of the predatory systems. Specifically, they found that those genes which encoded the proteins had all come from the same genes found in their non-carnivorous distant common ancestors, pointing to the fact that despite evolving at different times in different trap structures, the genes that encoded the digestive enzymes were only able to evolve from a certain set of genes found in all non-carnivorous plants. The phylogenetic tree shown was created in the study by comparing the genes responsible for the digestive proteins. The red lines on the family tree indicate where carnivory evolved. The green boxes indicate the 4 species that were compared, and the brown box shows the coffea plant used to compare it to follicularis' genes. Because there is a limited pathway or set of genes from which these digestive enzymes can be produced, the carnivorous plants convergently evolved similar digestive enzymes despite having evolved at different times and from different clades. The main reason these ancestral genes all seem to be encoded for carnivory is that in times of abiotic and biotic stress, these common genes specifically became more volatile and mutated to produce more effective proteins than that of which the past gene encoded. For example, the chitinase protein that the plant encoded to fight off fungal infection mutated slightly to make it effective at digesting the chitin in bugs.
Despite apparent differences in the carnivorous structures, the convergent evolution seen in the digestive enzymes shows that there are very few evolutionary pathways for plants to become carnivorous.